Hi everyone, welcome to chapter 5, Protein Function. In this chapter, we will explore how proteins interact with other molecules and how these interactions are related to dynamic protein structure. In this lecture, we will understand as to how proteins interact with other molecules. The learning goals of this chapter are as follows. We will first look at methods of binding ligands and proteins and understand how protein-ligand interaction works. To examine this further, we will look at quantitative and graphical modeling of protein-ligand interactions. And then we will look at some examples. We will look at interactions of globins with oxygen and non-oxygen ligands. Furthermore, we will look at physiological regulation of oxygen binding. And finally, we will look at three different mechanisms, beginning with the mechanism and control of antigen-antibody interactions. Second, we will examine the mechanism of muscle contraction and regulation of muscle contraction. In chapter 4, we looked at structures of globular proteins. In this chapter, we will examine certain functions of globular proteins. To be specific, one of the characteristic cellular function of globular proteins is to bind molecules. And a molecule bound reversibly by a protein is called as a ligand. Now, when a protein binds a ligand, there is always specificity involved, which means that the site where a ligand binds in a protein, which is called as a binding site, is almost always specific for a protein. For example, if I were to draw a structure of a protein, let's assume that this is a protein. A ligand can bind in this site, and so this is termed as the ligand binding site. Now, ligand binding is often coupled to conformational changes, sometimes quite dramatically. This means that once a ligand binds a protein, it can alter the structure of this protein, which means that once the ligand binds, the structure of a protein can change like that. That depends on the nature of the ligand and the site where it binds a protein. In multi-subunit proteins, conformational changes in one subunit can affect the others, which means that if you have a protein with multiple subunits, for example, if there is a protein that has 4 subunits, 1, 2, 3, and 4. Now, in this case, if a ligand were to bind in this site, this means that binding here can alter the properties of a site distant from this specific subunit, and this can make another ligand bind here, and that is how ligands can affect one subunit or binding of one subunit, affecting the binding of another subunit to bind another ligand. And this is termed as cooperativity, and we'll learn about this. Interactions can be regulated. This is illustrated by proteins like hemoglobin, various different antibodies, and proteins that control muscular movements. There are a variety of functions that are associated with globular proteins. The first one is storage of ions and molecules. And this is done by proteins called myoglobin. We have seen this before. Ferritin. Transport of ions and molecules. Hemoglobin is a characteristic protein that transports oxygen in blood. Serotonin transporter is another example. Defense against pathogens. Antibodies are one of the best examples that can that can initiate defense against a foreign body like a pathogen. Cytokines, another example. Muscle contraction. Actin and myosin are two different proteins that work in tandem to initiate muscle contraction. Biological catalysis. Globular proteins can also be enzymes. Two characteristic enzymes are chymotrypsin and lysozyme. The interaction of proteins with other molecules is almost always reversible. Reversible transient process of chemical equilibrium is given by this equation. A molecule that binds to a protein is called a ligand. It's typically a small molecule. But small proteins can also be ligands of other proteins. And as I said before, a region in the protein where the ligand binds is called as the binding site. Ligands bind via same non-covalent interactions that dictate protein structure. It allows the interaction to be transient.